Hello, we are going to start with chapter 4, Carbon and the Molecular Diversity of Life. So carbon is considered the backbone of life. All living organisms are mostly composed of carbon-based compounds. And carbon has the ability to form large, complex molecules, proteins, DNA, carbohydrates, and other molecules that make up living matter are all composed of carbon compounds. Therefore, what is organic chemistry? Organic chemistry is the study of carbon compounds, or compounds that contain carbons. Organic compounds are those compounds that are made up of a long skeleton of carbon atoms. So you will have a number of carbon atoms in a long chain that form the skeleton. And all living organisms, we contain a carbon atom skeleton. Therefore, we are composed of organic compounds. Opposite of organic is inorganic. So inorganic means there are no carbon atoms present. And we know that organic compounds will range right from simple molecules to colossal ones or complex molecules. Now, the major elements of life, as we learned in the previous chapter, in chapter 2, are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And I told you to remember the Chon group. Carbon, hydrogen, oxygen, nitrogen are the 4 basic elements. Now, in addition, we need phosphorus and sulfur, which we are going to see are uniform from one organism to another. And because carbon has the ability to form 4 bonds, these building blocks can be used to make a variety of other organic molecules. The great diversity of organisms on the planet is on account of carbon being a versatile atom or a compound. So carbon atoms can form diverse molecules by bonding to 4 other atoms. I discussed the electronic configuration. Electronic configuration is the arrangement, kinds and the number of bonds an atom will form with any other atoms. So we looked specifically at carbon. We need to know the carbon element had atomic number 6. Therefore, looking at its electronic configuration, how are we going to distribute the 6 electrons? First orbital could hold a maximum of 2. So first orbital has 2 electrons, 6 minus 2 was 4. So carbon atom has 4 valence electrons. And because it has 4 valence electrons, it can now form 4 covalent bonds with other atoms. This makes the large complex molecules possible. Now, in molecules where there are multiple carbon atoms, each carbon atom will be bonded to 4 other atoms. And if that carbon atom is bonded to 4 other atoms, it forms what we call a tetrahedral shape. Tetra comes from the word 4. So a tetrahedral shape. However, when 2 carbon atoms are joined together by a double bond, the atoms joined to the carbons will be in the same plane as that of the carbons. So we're going to see that in this slide here. So we take note of the molecule that we're referring to and the molecular structure, looking at structural formula ball and stick model. And we won't go into details with space filling model. In chemistry, you would be building up the models. In biology, we won't be building molecules. But take note of methane or methane, hydrogen bonds. So CH4 is referred to as methane. It is a gas. So you have the hydrogens. So carbon, and so this tetra, this is the tetrahedral structure. 1, 2, 3, 4. Ethane molecule has a molecular structure of C2H6. 2 carbon atoms, and you have each carbon atom bonded to 3 hydrogens. When you have CH3, it's also called a methyl group, which we'll be talking a little later. So you have 1 carbon atom bonded to 3, 1 carbon atom bonded to 3 hydrogens. So C2H6, and that's ethane. Now let's look at ethene or ethylene, which is a gas. So you have C2H4. But what do we notice? The difference between ethane and ethylene is, or ethene, is that there is a double bond between the carbon atoms. Yes, there was a single bond. And when there is a double bond between the carbon atoms, there is 1 less hydrogen. So you have just these 2 branch chains, CH2, CH2. So you get C2H4. 2 hydrogens less because there's a double bond between the carbon atoms, and therefore the gas is called ethene or ethylene. You see, the 2 carbon atoms will be in the same plane. So you have electronic configuration of hydrogen. To reiterate this information again, which we've covered in chapter 2, you see hydrogen has 1 valence electron, atomic number 1. Oxygen had atomic number 8. Therefore, first orbital 2, second orbital is 2, 4, 5, 6. It needed 2. It has 2 valence electrons. Nitrogen has 3 valence electrons and carbon has 4 valence electrons. So the electronic configuration of carbon gives its covalent compatibility with many different elements. And the valence of carbon atom causes the carbon atom to bond to hydrogen, oxygen, and nitrogen, as we saw. So they are the building codes for all other living molecules. So we see carbon dioxide, CO2. You have 1 carbon atom, it's forming double bonds with O. We looked at the double covalent bonds. Why? Because 2 pairs of electrons were being shared, because oxygen's 2. And so we see in carbon dioxide gas, the 2 oxygen, the valence electrons of oxygen are being shared with carbon. Let's look at the molecular and structural formula of urea. It's very important. You could be asked this question as a multiple choice question in the exam. So you have CO, NH2 twice. C double bond O, NH2. Whenever you see the NH2 group, that is the amino group. So you have 2 amino groups attached to C double bond O, which is urea. So urea consists of amino functional groups. Now, carbon chains form the skeletons of most organic molecules, and these chains can vary in their length as well as in their shape. So let us look at the length and the shape. So when you look at the length, these structural formulas are very important. You need to know the structure to be able to identify these structures in the exam. These are questions that could be asked. So let's look at ethane. Ethane was C2H6. So the number of carbon atoms are going to vary in the length. Let's look at propane. Propane has C3. There are 3 carbon atoms. So the molecular formula for propane would be C3 67678. C3H8 would be propane. Now let's look at branching. The carbon atoms vary in the length, meaning the carbon number of carbon atoms can increase. And here there are branches on the carbon atoms. So there's branching taking place here. So the difference now in butane. Butane is 1, 2, 3, 4 carbon atoms, and you have 3 and 3, 6, and 4, 10. So C4H10 is the molecular formula for butane. If you look at 2-methyl-propane, which is also called isobutane, how does it get its name 2-methyl? CH3 is a methyl functional group. So if you look at carbon atom 2, on the second carbon atom, there is a branching. There is another methyl group on the top of carbon atom 2. Therefore, we refer to this as 2-methyl-propane. Let's look at a double bond position in the carbon atoms. So you have 1, 2, 3, 4 carbon atoms, but on the first carbon atom, there is a double bond. Therefore, this is termed as 1-butene, but you should also take note that when there's a double bond between the carbon atom, there are 2 less hydrogens at the bottom. So this structure is called 1-butene. Let's look at 2-butene. So because the double bond is at the second carbon atom, this molecular structure is C4. You get 3, 4, 5, 6, 7, 8, C4H8. But remember, second and third carbon atom will lack 2 hydrogens. Now, some have the presence of rings. Some carbon atoms are in the form of ring structures. So if you look at a ring, now cyclohexane, hexa comes from the word 6. So you have 1, 2, 3, 4, 5, 6. But you notice that each carbon is attached to 2 hydrogens. So you'll have C6H12. Cyclo, associate the term cyclo with cyclic round structures in a circle. So cyclohexane, C6H12. What is the difference between cyclohexane and benzene? Benzene is a ring structure, but the difference is there are double bonds in the ring structure. Every alternate carbon atom has a double bond. And so you have to know exactly where to place the double bonds in the carbon atoms. And because there are double bonds, there will be only 1 hydrogen. So this is C6, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, C6H6, and benzene. So whenever you think of benzene, it's a ring structure and it has double bonds that alternate. Let's go to the term hydrocarbon. Hydro comes from the word hydrogen. So when hydrogen atoms are attached to the carbons, we refer to them as hydrocarbons. And hydrocarbons are organic molecules, mainly consisting of carbon and hydrogen atoms. Most of the organic molecules, such as fats, they have hydrocarbon components, and they can undergo reactions that are going to release large amounts of energy. So a classic example of the presence of hydrocarbons is in a fat molecule. If you take a section of fat tissue, you see the fat cells, part of a human adipose cell. Adipose comes from the word fat cells. This is a human tissue of fat cells. So you can see the fat droplets here. You can identify the nucleus of the cell. And this is the tissue of a fat cell containing the fat cells. What then does a fat molecule consist of? It consists of, these chains are called fatty acid chains. So it consists of 3 fatty acids and 1 glycerol molecule. So fat consists of 3 fatty acid chains with 1 glycerol. Now we look at the term isomers. Iso means same, isos. So isomers are compounds that have the same molecular formula, but they have different structural formulas, and therefore their properties will also be different. We have structural isomers. Structural isomers are isomers that have different covalent arrangement of the atoms. So when the atoms are arranged differently due to the covalent bonding, we call those molecules as structural isomers. And there are 2 structural isomers, cis and trans. Both put together are called cis-trans isomers. So cis-trans isomers have the same covalent bonds, but they differ in the way they are arranged. And that's what we call, they differ in spatial arrangements. Enantiomers are isomers that are mirror images of each other. So if you hold your hand in front of the mirror, this is my left hand, but it appears to be as the right hand. This is my right hand, it appears to be my left. So when you hold your left and right hands, they are like superimposed. They're mirror images of each other. So let's look at the structural isomers here. So we are looking at pentane. Hexa is 6, penta is 5. We have pentadactyl, meaning 5 digits, so 5 carbon atoms. 1, 2, 3, 4, 5. So it would be C5 molecular structure for pentane. C5, these are 2, 4, 6, 8, 10, and these 2, C5H12 is pentane. Look at the structure of pentane and 2-methyl-butane. If you look at 2-methyl, why are we calling it 2-methyl? Because you have, at the second carbon atom, you have a methyl group. So you have C4. You have this methyl group here. So this is 3 hydrogens, 4, 5, 6, 7, 8, 9, 10, 11, 12. So 2-methyl-butane. Structural formulas are different. The molecular formula too. Let's go to the B component, cis-trans isomers. Cis isomer, you can see these X's. You have a double bond between the 2 carbons and hydrogens. So when the 2 X's are on the same, cis means same side. Trans means opposite. So if you were to rotate the carbon atoms, now these X's are on the opposite side. So trans isomer, you notice that the two X's are on opposite sides. So this, you rotate this carbon atom, this hydrogen goes up, X comes down. This hydrogen goes up, this X comes down. So they are on opposite sides of each other. So X's are on opposite sides of each other. So that's a trans isomer. So this was just an animation to show you structural isomers, geometric isomers, see, like cis and trans isomers, where it is on the same side, cis isomer, trans opposite sides. And in enantiomers, they were mirror images of each other, left and right hand. And you can clearly see this enantiomer over here, where you can see the red color is on the left, here it's on the right, here it's on the right, here it's on the left, it's rotated. So where do we use enantiomers? They are, or where are they important? They are important in the pharmaceutical industries. Two enantiomers of the same drug will have different or opposite effects. And only one of the enantiomer or isomer is normally biologically active. Differing effects of the enantiomers will show that the organisms are sensitive to even slight, subtle changes in the molecules. So we are looking at, we're going to compare two drugs. What effects do these drugs have in humans or in our bodies? Out of these two drugs, which one of these is the effective enantiomer? One is effective in the pharmaceutical industry because they have pharmacological effects, and one would be the ineffective drug. So we all are familiar with the drug ibuprofen. When do you take ibuprofen? When you have fever, when there's inflammation, to reduce inflammation and pain. And so you have two enantiomers. In this case, the effective enantiomer is S minus, is the S1, and the R dash ibuprofen is the ineffective one. So for example, if a pharmacist dispensed off a wrong drug, when they made the medication, by mistake, they dispensed all our ibuprofen. The S1 was never made, and it was sold to the pharmacies. What would happen? You would still have your fever, you would still have pain, your body would still be inflamed. There is no reaction taking place until someone could have detected that something went wrong. And that's how now you notice that many of the drugs are sometimes drugs are being recalled because of contaminations that are taking place. Could be a bacterial contamination. And therefore we see that the difference between this R and the S ibuprofen is that these stereoisomers, they are very similar in their properties. Look at the structures, they're very similar in their properties because they have the same melting point, boiling point, as well as the solubility. But we notice that the S plus form is more pharmacologically active form. The R1 has no anti-inflammatory effect. And so it does not reduce or fight against inflammation, thereby not reduce the inflammation or the pain that the individual may be suffering. Let's look at albuterol. When is albuterol dispensed or given to an individual? If you're asthmatic, In asthmatic patients, they give you albuterol. Why? Because it relaxes the bronchial or the airways, the muscles, respiratory muscles, the airways, and thereby allowing the individual to breathe because there is more flow of air. In this case, it is the R-albuterol that is the effective enantiomer and not the S. So take note of these two. Now, what happens in this case? This R-albuterol, it is going to bind to the beta-2 adrenergic complex receptor with greater affinity than the S-albuterol enantiomer, and therefore it is responsible for albuterol's bronchodilating activity. We don't need to remember, but this is good knowledge. And so, therefore, the individual would be able to now breathe because there is more airflow through the respiratory tract, and the bronchioles have now opened up the air sacs. Okay, so R-albuterol is the effective enantiomer for albuterol, and here it is the S-ibuprofen, which is the effective enantiomer. Take note, very important slide, you could be asked a question. And here with the same thing, there's an animation of L-DOPA. Now, L in biology, we just call it L and D, but in chemistry, they actually tell you L stands for levo, D stands for dextro, left hand and right hand, like in the enantiomers, mirror images of each other. And in this case, L-DOPA, which stands for levodopa, L-E-V-O-DOPA. Levodopa actually is a prodrug that is being converted to dopamine, the neurotransmitter or the hormone, by an enzyme that can cross the blood barrier. This is what L-DOPA drug does. Now, the chemical groups that are more important in the processes of life, there are many sex hormones, but estradiol and testosterone are both steroids with a common carbon skeleton, which is in the form of 4 rings that are fused, they are joined together. And these are both sex hormones. Estradiol, estrogen. Estradiol is a derivative of estrogen, the female sex hormone. Testosterone is the male sex hormone. And both these sex hormones are going to differ in the chemical groups that are attached to the rings of the carbon skeleton. Take note, this is very important structures to both female sex hormone and male sex hormone. You could be given these structures and you will be asked to answer questions on these 2 sex hormones. So look at the difference that occurs. Yes, you have a methyl group and a hydroxyl group here. Same structure, 1, 2, 3, 4. You have the fused ring structures here. But what you notice is there is a methyl group attached here in testosterone, but here this benzene ring, this is a benzene ring, this is not a benzene ring. Here you have a double bond, O, and here you have hydroxyl group, OH. So look at the difference between the functional groups that are attached that helps you to identify which is the female sex hormone and which is the male sex hormone. And all these sex hormones are steroids in the body. Okay. Now, there are 7 functional groups that are very important in the chemistry of life. Hydroxyl group, carbonyl group, carboxyl group, amino group, sulfhydryl group, phosphate group, and methyl group. Let us look at different examples that contain these functional groups. So you must know, when you see the word OH, and we learned this when we talked about water dissociates into hydrogen ions, acids, and bases, which were hydroxide ions or hydroxyl group. So OH group is the hydroxyl group, OH. Where would you see hydroxyl groups? In alcohol. Example would be ethanol. So the moment you see an OH group, you must be able to identify that it is the hydroxyl functional group, and it would be seen in ethanol. The moment you see the suffix OL, associate it with the alcohol. Carbonyl group, C double bond O. Carbonyl groups are present in both ketones and aldehydes in carbohydrates. We'll be doing this in the next chapter when we talk macromolecules. So acetone, propanol, they are ketones and aldehydes. Carboxyl, carboxyl is COOH. So carboxyl group is present in an acid. An acid is COOH or carboxyl group. So acetic acid, CH3COOH. Acetic acid is none other than vinegar. Let's look at the NH2. We just saw in urea, 2 NH2 groups with C double bond with CO. So you have NH2 is the amino group. It's an amine, it's an amino group. Glycine, these are all names of amino acids, and proteins are composed of amino acids. Let's look at the sulfhydryl group. Sulfhydryl group is SH. They are thiols. Cysteine, you'll see a SH functional group, which shows you that it is sulfhydryl. Phosphate, PO4, the phosphate group, organic phosphates. Glycerol phosphate, we'll be seeing it in ATP molecule. And then methyl, we have to look at all these different molecular formulas which show the structure of the methyl group CH3. So you'll see it as methyl cystosine. So in particular, I want you to remember these functional groups. Okay, I won't ask you to memorize the structures of these examples. Just talked about phosphate, ATP. ATP is an important source of energy for cellular respiration. Take note of ATP because we'll be going into details when we look at cellular respiration, the chapter on when we cover cellular respiration. So what does ATP stand for? A stands for adenosine, T stands for tri, 3, P stands for phosphate. So the molecule has 3 phosphates attached to it. ATP is going to store the energy of a cell. It is the energy molecule, the energy currency of a cell, and it will store the potential energy. It has the potential to react with water. This reaction releases the energy that can now be used by the cell. So we see here adenosine, tri, 1, 2, 3, phosphate, PO4 is the phosphate functional group. PO4, PO4. So adenosine triphosphate is ATP molecule, which is the energy currency of the cell. And when our cells are performing any activities, or we humans are performing any activities at all times in the body, our cells are using this ATP molecule, which is the energy molecule of our cell. So This ATP molecule, adenosine, when it reacts with water, we notice that one phosphate bond gets broken down. So when one phosphate bond gets broken down, two phosphates remain. So therefore, that molecule is called adenosine diphosphate. Now, when this phosphate bond is broken down, this phosphate is organic phosphate. So it gets converted. The bond now is broken down, and that phosphate group gets converted into PI. I stands for inorganic phosphate. So you'll have A, P, P plus PI, and you'll have energy being released. The same thing, ADP now, one P can be broken down, you're left with one P and plus PI. One P will be adenosine monophosphate, and then energy is released. So it would be ATP gets broken down to ADP and AMP. We'll see this happening during cellular respiration. So this is the end of this chapter, students. I encourage you to go through the readings, look at the PowerPoint slides that I'm attaching in the folder. So every chapter with the plain slides, as well as another attachment with my narration. Take care, students. See you later. Bye-bye.